The Research, Part 1. We've been looking at the book Undeniable by Douglas Axe, How Biology Confirms Our Intuition That Life is Designed, published this year by HarperCollins. And a question arises, if you read the book, where does Axe get the confidence to say that we should believe our design intuition and ignore the scientific consensus? Well, at least part of the answer comes from a series of 4 papers. There was more, and we'll discuss that later, but these 4 kind of happen to be together and form a nice unit to get started on Axe's research. The first one is published in 1996 with N.W. Foster and A.R. Furst. The next one is also with Foster and Furst in 1998. And both of those, of course, are available on the internet, as are the other 2 papers. So you can check to see whether I'm presenting them accurately and fairly. The third one is also with Foster and Furst. All of these done at Oxford. Maybe Cambridge. I may be wrong on that. In fact, it is Cambridge, I think. And then then finally, there's the piece that he wrote alone in 2000. And I think that part may become significant later on. Well, before I do that, so that you're not totally lost, I should probably explain about the enzyme barnase. Barnase is bacterial RNase, which is to say it's an enzyme that's put out by bacteria to chew up RNA. And it's basically secreted outside the cell normally. And the idea is if there's a bunch of RNA out there, maybe some viruses the bacterium doesn't want, or maybe just food that it wants to break into pieces small enough for it to manage inside, and it doesn't want the RNA activity. It breaks it up before the pieces ever make it inside the cell. It's found, among other places, in E. coli. And as you can appreciate, it would be toxic to the secreting organism because before it gets exported, it would start chewing up RNA inside the cell. And in fact, if it's all by itself, that's what happens. The cell dies. So in order to stop that, inside the cell, it is associated with a suppressor or blocking protein, if you like, called barstar. And they form a hand and glove arrangement that is so tight that the constant of dissociation is something of the order of 10 to the minus 8. Very, very small constant. They tend to stay together very well. And you may have noticed in the background, what we have here is the barnase, and over here we have barstar fitting into it. And then, of course, once they get outside, they are enabled to separate, and then the barnase can do what it's supposed to do. But inside the cell, it has to have that combination. Well, now, in the experiments that were done, plasmid DNA was prepared with 2 substitutions for in barnase itself. Codons 28 and 57 are changed from serine to amber, which is a stop codon. Now, amber is not a well-used stop codon, and some E. coli strains actually don't recognize it as amber and stop making the protein, they instead put on serine. And there is an amber-suppressing strain of E. coli that will read these stop codons in serine, and most of the time, even when you have changed the DNA, it will put out a regular old protein anyway, which of course is fatal to the organism if it doesn't have bar star. And these particular E. coli also are deficient in barnase and bar star themselves. They can still live off of the rest of whatever the media they're in. They're disadvantaged if they're in human guts, but which is where most E. coli likes to live. But if you feed them enough other stuff, they'll do just fine in culture. The E. coli that's there is very, very sensitive to this protein, and that is to say that if barnase is being produced with 0.02, in fact, later literature says 0.01% of the normal activity, that is 1/10,000th of the normal activity, the strain will kill the E. coli if there isn't a suppressor. And they're choosing cells that don't have a suppressor, as well as cells. Now, some of them will be able to read amber as stop and not make this protein. Other ones will read the amber as serine, and they'll make the protein and kill themselves. Now, the plasmid also conveys chloramphenicol resistance and ampicillin resistance, but the ampicillin gene, the penicillinase gene, beta-lactamase as it's called, is actually has one of its serines chained to an amber as well. And so it won't make beta-lactamase unless it is in one of these amber suppressing cells that misreads amber as serine. So, if you have a strain that did not pick up the plasmid, you can kill it with chloramphenicol because E. coli normally does not have resistance to chloramphenicol. It's an antibiotic that we kept using a while ago because it could sometimes kill people. Rarely, but the people who died didn't think that the rareness mattered too much. Some of us who've been around for a while remember when it was actually used. And it is still used in people who are desperately ill, whose chances of dying from the disease are higher than their chances of dying from the chloramphenicol. But you're taking a calculated risk whenever you use it. Any strains that lost their amber suppression can be, with this plasmid in place, can be killed with ampicillin because they don't make the beta-lactamase. And if the remaining cells survive, they obviously have a plasmid, amber suppression, and less than 0.2% of barnase activity. Now, in biochemistry, we don't like negative assays. It worked if nothing grew. That's kind of a difficulty. So in order to fix that, you have to have controls. And the control is an E. coli that doesn't have barnase, or preferably doesn't have barnase, but also does read amber as a stop codon. Those cells will now, if they pick up the plasmid, they will also have chloramphenicol resistance. So if you want to get rid of the normal cells, you give them chloramphenicol, and then all that's left is the ones that picked up the plasmid. Okay. They don't have resistance to ampicillin because of the stop codon. So they can be killed with you can take some of them and kill them, and then the other ones, well, you still have that. They still have the plasmid in. And they don't have barnase activity because of the stop codons. And so therefore they'll grow well in chloramphenicol media, but you can kill them with ampicillin. So those are kind of your controls. Now, there is, well, of course, after you've grown them, you can then sequence the plasmid regardless of whether the barnase is active, because they don't translate the barnase for themselves anyway, because of those stop codons that are put in there just for this purpose. Now, there is one flaw that exists in this scheme, and that is perhaps amber-suppressing E. coli simply don't pick up the plasmid at all, and then they're killed by the chloramphenicol. And how would you know that they did that rather than being killed by the barnase? Again, that's why negative results are not, usually something you really like to use as a sensitive assay. But to be fair, the wild-type E. coli pick up the plasmids, which are not fatal to them, and the amber-suppressing E. coli pick up the non-fatal plasmids. So this, while possible, does seem unlikely. And how unlikely? Well, if you get 300 colonies on the average of the other ones after you give them chloramphenicol, that works out to e to the minus 300 that you would get no colonies, which is 1.9 times 10 to the 130. So it's kind of unlikely. I think that's a significant p-value. Okay, so let us get started now that we understand a little bit about the assay that's being used. The central structural feature of natural proteins is a tightly packed and highly ordered hydrophobic core. This is well known from proteins. They have parts that avoid water that cluster together. They have parts that like water that are out floating around, in water. And that's how they make their shape. If some measure of exquisite native-like core packing is necessary for enzymatic function, so that all those hydrophobics have to fit in just the right way, this would constitute a significant obstacle to the development of novel enzymes, either by design or by natural or experimental evolution. So it would be hard to make proteins in that case. To test the minimum requirements for a core to provide sufficient structural integrity for enzymatic activity, we have produced mutants of the ribonuclease barnase in which 12 of the 13 core residues have together been randomly replaced by hydrophobic alternatives. It actually turns out they did 13 as well, as you will see. Using a sensitive biological screen, we find that a strikingly high proportion of these mutants, about 23%, retain enzymatic activity in vivo. Further substitution at the 13th core position shows that a similar proportion of completely random hydrophobic cores supports enzyme function. A little bit less, which is, so it's probably more like about 20%. Okay? Of the active mutants produced, several have no wild-type core residues at all. These results imply that hydrophobicity is nearly a sufficient criterion for construction of a functional core. Well, not really nearly. It's a chance in 5, but it's a pretty significant chance. And in conjunction with previous studies, the refinement of a crudely functional core entails more stringent sequence constraints than does the initial attainment of crude core function. So the idea is that you can get this enzyme started with an almost random. Well, if you do it 5 times with a random inner core, which is amazing, was amazing to everybody when it first came out. Now, since attainment of crude function is the critical initial step in evolutionary innovation, you have to have some function before you can refine it. The relatively scant requirements contributed by the hydrophobic core would greatly reduce the initial hurdle on the evolutionary pathway to novel enzymes. Similarly, experimental development of novel functional proteins might be simplified by limiting core design to mere specification of hydrophobicity and using iterative mutation selection, that is, standard old evolution, to optimize core structure. The paper begins, tight, well-ordered packing of interior hydrophobic side chains plays an important part in stabilizing the unique active conformation of natural proteins. I thought that was what they're going to prove it didn't require. Well, this is what was thought before the paper. Introduction of even small changes to a hydrophobic core typically diminishes the packing quality. Oh, so they're going to dispute this, the stability and often the activity of the protein. Well, actually, kind of. You see, they're not saying that these things don't, that they all have as good as the wild type activity. What they're saying is that they have enough activity to where you can start selecting for it, selecting for more and more activity. Reproduction of such exquisite core packing in proteins designed de novo has proved to be a considerable challenge. It's tough to make it. Well, as we're going to find out, it's not quite that tough in the case of barnase. If a design fold lacking an engineered core exhibited even slight activity, this might serve as a starting point for experimental evolution of a highly active novel structure. Highly functional natural proteins provide an opportunity to test the feasibility of this approach. Is it possible then for a protein completely devoid of native-like core packing to exhibit phenotypically detectable activity? Of course, their answer will be yes, but to tackle this question, we have saturated the core of barnase with random hydrophobic substitutions and applied a very sensitive biological screen to detect activity in the resulting mutants. I'm not going to go over exactly how they did that. It involves making plasmids and mutating certain sites and so forth. And you will notice that although the outside core has phenylalanine, valine, alanine, leucine, tyrosine, that there's no tyrosine in the substitutions because of the way they did it. And there's no alanine in the substitutions because of the way they did it. And then there's also isoleucine, more tyrosine. There is a tryptophan here and a couple of more isoleucines. That's the original type. That's the kind that 99 point something % of E. coli have if they have this enzyme. You will notice that the stage 1 mutants, they've mutated the first 6 places. That's because they're all from 7 to 24 and they're kind of all in a group. The next ones are 74 to 96, and then they did the 109 later, you'll see. But you can see that from 74 to 109, they're all identical. So the first stage, they're just mutating the first 6. And you can see that a little over half of them have some activity. Now, some of them have a little less activity, but you can see that clearly there's a lot of them that you just mutate the first 6. Doesn't matter what you put in. Okay, well, what happens if you mutate the second? Now they've mutated everything except for that final isoleucine. And by the way, I don't know how they did it, but somehow when they did that, these ones wound up mutating as well. So there is none of these that match the first 23. I know, I looked. It was a pain in the neck. But now you can see that there's a lot less. And there's more than the 42 there. It actually goes to 59. It actually goes to 75. So they had a whole lot of them that they did. And you can see that, probably about a quarter of them, a fifth of them, something of that nature, excuse me, actually have activity. Okay? So, then they did the third stage where they mutated the isoleucine. Now, this is interesting in a number of ways because they did this by itself. They actually picked number 34 and mutated it two different ways. They picked number 40 and mutated it two different ways. Basically, they just tried to mutate it, and this is how it came out. They didn't directly mutate it and say, oh, I want a leucine here and I want a phenylalanine there. That's just how it came out. As they got better, they are able to be more specific about the precise mutation they want. But as you can see, there's a lot of them that are and a lot of them that aren't, but you'd like to know which ones of these actually had activity before they mutated the isoleucine. Well, I can tell you that these ones did not have activity to begin with, and you'll notice that they didn't have activity afterwards. So apparently, mutating an isoleucine isn't enough to turn it back to activity. There's another interesting point. This one, 63, is actually a 1+. And you'll notice that if you mutate to leucine, it gives you 2+. So you can gain a little bit of extra activity that way. On the other hand, 68.1 is a 2+ that went to a 1+ when the isoleucine was mutated to a leucine. So, you can get gains and losses in activities, but apparently nothing goes to from full plus to complete minus, except for 3 of them. And interestingly, those 3 all had phenylalanine substituted for the isoleucine. So there they are. And if you take those ones out and you take the ones that didn't have any activity to begin with out, everything else has activity, suggesting that you really don't want phenylalanine in your as your last amino acid. Thus, the results presented here demonstrate that mere hydrophobicity is nearly sufficient. Again, we're being a little bit exaggerated. It's only 1/5, but hey, it's pretty close. As a criterion for the construction of a barnase core that provides adequate structural integrity for enzymatic function in vivo. Of all possible amino acid combinations at the 13 core positions, approximately 1 in 3 times 10 to the 4th would fully satisfy the hydrophobicity criterion. That is, if you just mutated them, whatever. Why is it so low? Well, it's because only 9 of the 20 amino acids get to count as hydrophobic. See? And you'd have to have every single one of those be hydrophobic, which is 9 over 20 to the 13th power, which is, a little less than 10 over 20, or 1/2 to the 13th power, or which figures out to 1 in 3 times 10 to the 4th. So you're not likely to get it, but if you have billions and billions of bacteria, you could. Here we find that approximately 1 in 5 core combinations meeting this criterion, or approximately 1 in 2 times 10 to the 5th overall, supports enzymatic function within the cell. Hence, whatever further criteria may be needed for sufficient specification of functional barnase cores, they accomplish only a 5-fold reduction in the number of core sequences. That is, 1 in every 5 is correct. And now, how do you get better than that? I don't know, but you can't get too much better than that. That the requirements for attainment of a functional core are relatively lax has important implications for protein evolution. And remember, Doug Axe is writing this along with 2 other really sharp people who are, as we found out in the book, one in particular, Fecht, is a convinced mechanistic evolutionist. So he's liking this. Yes, this is what we're going to say. The considerable optimization power of biological selection requires a pre-existing structure exhibiting a phenotypically significant level of activity. The presence or absence of such starting structures is therefore critical in determining the evolutionary course of any system undergoing replication with random mutation. Now, this my comment after the paper. Note that this research superficially favors an evolutionary pathway. Also note, in a related, possibly a point, that 2 of Axe's fellow researchers were quite willing to put their names on the paper. Also note that while it makes explaining the existence of some barnase, some kind of barnase, easier, it does not explain, it makes it harder to explain, why virtually all barnase in E. coli has the same sequence, which is why it's called the wild type. Well, maybe that's the very best, but then there are other bacteria that have another one. Why not have mixtures of the 2 or the 3 or the 20 or the 500 that all work? Or what, the 10 to the 15th that all work? I mean, it's a huge number of different kinds of barnases. I guess it would be 9 to the 13th power, but it's a huge number of different kind of barnases that could theoretically be working, and maybe 3/4 of them are below the level, and so you'd like to get high, but there's a huge number of barnases that could have been working in E. coli. So why does all E. coli have 1? Well, there's a bottleneck somewhere. But then why after the bottleneck? Because humans have been around for, thousands of years at least. And E. coli loves humans. Humans don't know enough to love E. coli most of the time, but they all have it. And so why do they all have the wild type? Or, 90-some% of them have the wild type anyway. It looks like we have a monoclonal situation. And that's especially weird if you think that we've had 10,000 humans going for 6 million years. You would think there'd be some divergence because there's a whole bunch of different barnases that would work. Why do we have only 1? So this research superficially supports evolution, but in reality it doesn't. What it does is ask the question, if neutral evolution can occur, why isn't there more of it? Okay. We move on to the second paper. Exhaustive substitution studies where many amino acid replacements are individually tested at all positions in a natural protein have proved to be very valuable in probing the relationship between sequence and function. The broad picture that has emerged from studies of this sort is one of functional tolerance of substitution. Now, this is doing individual substitutions, okay? We have applied this approach to barnase, 110 residue bacterial ribonuclease. Because of the selection system used to score barnase mutants as active or inactive, detects activity down to a level that can be approached by non-enzymatic catalysts. You might as well not have the protein. Mutants that test inactive are essentially devoid of enzymatic function. Of the 109 barnase positions subjected to substitution, there's actually 110, but one of them they didn't bother with, so one on the end, as you will see. And only 2 could not be substituted without such inactivation. A total of 33 substitutions, amounting to 5% of the explored substitutions, were found to render barnase wholly inactive. The profoundly disruptive effects of all of these inactivating substitutions appears to result from either 1, replacement of a side chain that is directly involved in substrate binding or catalysis. It's the ones that actually do the job, or that line up the RNA so it can do the job. 2, replacement of a substantially buried side chain that's holding things together. Not just a non-specific side chain as we saw from the last paper. 3, introduction of a proline residue, which kinks the protein at a specific point. Or 4, replacement of a glycine residue, which we'll have more fun with later. Although substitutions of these types are functionally tolerated more often than not, the system used here indicates that only these sorts of substitutions are capable of single-handedly reducing catalytic function to or nearly to levels that can be achieved by non-enzyme catalysts. That is to say, you don't destroy completely the function with one substitution, with a couple of exceptions. It is hoped that investigations of protein folding will ultimately yield a comprehensive understanding of the relationship between protein sequence and structure. And this is the beginning of the paper. Although this is an ambitious undertaking, it is only half of the larger effort aimed at elucidating the relationship between sequence and function. What difference does it make if you change 1, 2, 5, 10, 20 different amino acids? The other half, aimed at understanding the structure-function relationship, is no less ambitious. While a general solution to the grand problem of relating protein sequence to functional will clearly be some time in the making, we do have at our disposal, in the form of natural proteins, thousands of specific solutions to this problem. We don't know how to make proteins, but we can look at what we've got in terms of enzymes that actually function. It therefore makes good sense for us to glean as much raw data as we possibly can from these natural solutions. Given the complexities and subtleties of the sequence-function relationship, it also makes sense for us to collect these data in a manner that is entirely unbiased by any a priori expectations we may hold regarding the nature of that relationship. You like to not cherry-pick your data. And the easiest way to not cherry-pick your data is to gather all the data. The simplest and most direct way to obtain such an unbiased data set is produce and test a large collection of mutant proteins where all possible single residue substitutions are represented. You collect all the cherries, and then you're not cherry-picking. Okay? Since all single replacement possibilities are examined by this approach, the data lack any imprint of the experimental's expectations, and they are complete in the sense that the entire molecule is examined. A number of studies have captured the essence of this approach, and there's references 1 through 8, of course. There is a positive correlation between the degree of solvent. There, I shortened the word slightly, and so you need it for the English. Between the degree of solvent exposure at an amino acid position and the level of substitutional tolerance at that position. It has also become clear that natural proteins typically tolerate single substitutions, even non-conservative ones, at most positions without complete loss of function. Barnase, our friend, provides an opportunity to apply the exhaustive substitution approach using a very low activity threshold. The extreme autotoxicity of this enzyme allows direct selection of mutants with very low, approximately 0.1% of wild type and lower. Notice it's gone down lower than the 0.2 activities. 12 is Douglas Axe, what's his name in Furch, the or first the previous paper that we talked about, thereby enabling us to directly identify residues that profoundly influence enzyme function. So he's building on his previous paper. And I'm not going to go into all the details of how they did it, but I am going to give you the results they have. And you will notice here that most of the time it'll tolerate anything that they do. Now, they haven't done everything. As you can see, there's somewhere between 5 and 7 different amino acids that have been substituted. And this is going through and just substituting a single nucleotide. That's why you don't see 20 of them, or more precisely 19, because they didn't actually try doing double nucleotide substitutions. So that, for example, everywhere you see, let's say arginine, which is this one, you have CSGLPH, even where it doesn't work. Every time they do arginine, that's the 6 amino acids that they test. And there's some that it's easy, for example, glutamine will have 7, and here this tryptophan will have 5 that it can easily be translated into. So they systematically go through and they change one nucleotide at a time, and they see what. So it's not perfectly 20, but it's, it's pretty indicative. And the thing that's interesting is, here's one. If you start with tyrosine and you change it to aspartic acid, you're in trouble. But if you change it into serine or histidine or glutamine or phenylalanine or cysteine, doesn't matter. They all work. Same way with leucine, you change it to arginine, you're in trouble. You change it to any of the other ones, you're fine. Proline, as you will notice, kind of pops up every once in a while. And you can do anything but proline. There are 2 that you can't substitute anything for. Well, we don't know for sure. This is arginine, and maybe if you substituted lysine for it, you could get away with it. They didn't try that, actually. But you definitely, most of the amino acids won't work there. This is interesting because arginine is a positively charged side chain, and that would make sense if it was trying to line up negatively charged ribonucleic acid. And in fact, both of these arginines, you see the underlines happen to actually touch the RNA. Here's another one. This glutamic acid also touches the RNA, but it doesn't seem to matter. The serine here doesn't seem to matter either. The only other ones here are, well, let's say aspartic acid, and you can get away with glutamic acid, or asparagine, or glycine, but you can't get away with histidine, valine, tyrosine, or alanine. So, that's what it says, is it kind of related stuff, or maybe nothing at all you can get away with, but not, it has to be related, otherwise it won't work. And here, histidine, again, you can get away with leucine or phenylalanine, but you can't get away with glutamine, tyrosine, asparagine, aspartic acid, or arginine. That has to be histidine. And that's interesting because both arginine and histidine are positively charged. So apparently, you can't go from one positively charged to another. It ruins the enzyme. Anyway, but as you can see, outside of these arginines and this histidine, and maybe this glutamic acid, pretty much you can do whatever you want to. Now there's some weird stuff here where glycine goes to valine, and right here, and we're going to come back to that in another paper, so that's why I'm pointing it out to you. You'll notice that if you put valine in either of these places, it won't work. But there's a tremendous amount of variation that you can do. You can do single amino acid substitutions all over the place. And like 95% of them will work. Among the interesting class 3 substitutions are the ones that replace either of the two glycines at position 52 and 53. Those are the ones I pointed out to you. At both positions, valine is seen to be inactivating, but a number of non-conservative substitutions apparently do not completely eliminate activity. Valine's the only one that really does. Now, it'd be interesting to see, probably isoleucine would also not work, but, you know, because valine and isoleucine are very, very closely related. So there might be a few more that won't work. But valine and isoleucine are both the two bulkiest amino acids. And they're both hydrophobic. So apparently glycine in that area does not do well when those are substituted. Including alanine, by the way. The fact that numerous independent examples of the valine substitutions were recovered, 7 examples of G52V and 4 examples of G53V, suggests that most of the mutants that were not recovered really are active. They were able to show that the plasmid actually had that substitution in it. The presence of a beta bulge, a beta sheet distortion caused by surplus residue in one strand, involving residues 53 and 54, raises the interesting possibility that this small structural element may be important for barnase function. The results of a detailed investigation of the effect of substitutions in this region will be reported elsewhere, which we will get to next. Design implications. Whoa, how they snuck that into the paper, I don't know. You know, you can't talk about a designer anymore. But I guess this was before it was verboten. In light of the above discussion on the significance of essential features, we should now consider what the results of the study imply for the design of a barnase-like enzyme. For the reasons indicated previously, we must here focus on these features that appear to be essential, recognizing that the list of these will probably be incomplete. The first implication to consider is that any protein design aspiring to emulate the fold, and there's that word design again, and mechanism of barnase will need two arginines to fill the roles of R83 and R87. Those are the ones that didn't substitute. You got to have that. And probably a histidine to fill the role of H102 as well. That's the one that only 2 other substitutions worked for. H102 does not appear to be truly essential, but it is sufficiently sensitive to substitution to suggest that it may be essential in anything but a highly optimal context. Studies involving combined substitutions, as in reference 12, will provide a clearer picture of the sequence requirements for basal barnase function. This work constitutes an essential first step as it provides information that is needed for the design of multiple substitution experiments. So he's looking on, well, let's see what happens when we do more than one. Single amino acid residue substitutions rarely destroy the structural integrity of proteins. You'll notice that it only did that in less than 5%. Substitution of glycine residues, however, is among the few sorts of alterations that can have such an effect. Here we seek to understand what accounts for the extreme... We have now moved on to our 1999 paper. And so they're going to pay attention to that little glycine area. Here we seek to understand what accounts for the extreme functional impairment of the bacterial ribonuclease barnase upon substitution of glycine 52 or glycine 53. We find that inactivation is caused by overall disruption of the folded state that manifests itself in 3 ways. By 1, dramatically reduced stability, and that's a measure of how unstable it is. Progressive loss of folded to progressive loss of folded state activity with increasing temperature, indicating a less well-formed fold. That is, as you heat it, it gets more disordered and it doesn't do what it's supposed to. And 3, substantial proteolytic degradation of mutant enzymes in vivo. That is to say, it doesn't work as well in the cell as it does in the test tube, because in the cell there's these enzymes waiting for proteins to unfold, and as soon as they start doing that, they chop them up. Examination of 2 deletion mutants missing either glycine 53 or asparagine 54, which is where there's the extra one, as we will see, shows that the irregular B bulge formed by these, that should be beta bulge. I missed that. You can see it at the bottom, I caught it. Formed by these 2 residues is of vital importance to the structural integrity of barnase. The parallel behavior of mutants carrying replacements of either of the 2 glycine residues therefore appears to arise from a common mechanism, disruption of local structure at the beta bulge. The importance of this structural element to the function of barnase raises the question of whether it may be present in other RNases. The streptomyces enzyme RNase SA and RNase ST, which are 2 different streptomycin enzymes, differ considerably from barnase in both sequence and structure, yet both show significant sequence similarity to barnase over a region beginning at, for barnase, Gly53. So they're saying that this is not just true for barnase, it's true for other RNases that aren't barnase, definitely aren't barnase. Structural comparison indicates that the streptomyces enzymes do have the barnase-like irregular beta bulge, making this an important characteristic feature of a group of bacterial ribonucleases. The sensitivity of this feature demonstrates that detailed aspects of local structure may have a major role in determining the overall structure and functional properties of an enzyme, even when no explanation for this role is readily apparent. It's important. We don't know why, but that doesn't mean it isn't important. If this is a general characteristic of the structure-function relationship, it may pose a formidable obstacle to the de novo design of new enzymes. You have this huge thing, and it's all dependent on one little bulge. How are we to know that? And how are we to know whether the next enzyme doesn't have a little deformation there that's necessary? Introduction. For over a decade, protein engineering methods have been applied with remarkable success to the study of protein function and its relationship to protein structure. And here, First and Winter, First, remember, is the 3rd author of this particular paper, and Gurlt in 1994. The rapid growth of this field following its inception can be attributed in large part to the vast and rapidly growing database of known protein structures. It's nice to know what proteins look like if you're trying to figure out how they work. An alternative approach, which would be termed the function-led approach, is to employ a biological screen or selection procedure to score randomly or systematically generated protein variants as either active or inactive. This approach offered several advantages, and this is the approach of this paper. We're going to mutate it and see what happens. And for what it's worth, this is only part of the beta sheet. There's some more of the beta sheet out here, as you can see these dotted lines that go out to, in this case, hydrogens, and in this case, oxygens, from another sheet that keeps going. There's actually 4 of them, as we'll see in a little bit. But you can see this thing has a nice regular structure, except for right there, where you have 2 amino acids instead of the one that should be there. And that forces it to kind of do a little up and down, loop-de-loop, and then come back to normal. Here's another drawing of it. And you can see that this is NH, and there's the O that's bonding up. And actually, I think this is a nitrogen, and that's the carbon, come to think of it. But you can see how they're bonding together. The hydrogens are just left off. And And as you can see, it kind of bends up like that. Instead of being nice and flat, you have this little doodad that sticks out. They're called a beta bulge. I'm not reading the whole paper, obviously. I'm just trying to give you the most important parts of it. Our analysis focuses on 6 barnase variants. Mutants, of course, the original. Then mutants G252V and G53V, which are the ones they created in the last experiment, were chosen for study because, as mentioned above, they are known to be essentially devoid of enzymatic activity in vivo. And there's the reference to the previous paper. And our primary objective here is to elucidate this phenomenon. The previous study found that the following amino acid residues are, unlike valine, tolerable substitutes for either of the 2 glycine residues: alanine, cysteine, serine, asparagine, and arginine. So they're going to particularly look at alanine. Of this set, one would expect alanine to cause the least disruption because it most resembles glycine. Since alanine is also intermediate in structure between glycine and valine, we chose to include the 2 alanine mutants G52A and G53A, which work, remember, in the study. Finally, 2 deletion mutants missing either glycine 53, or for that matter could be glycine 52, because they're equivalent if you have a deletion, or aspartic acid 54, which is D54 delta, D54 minus, if you like, are included so that the effects of removing either of the 2 residues forming the beta bulge can be determined. So they're going to, we'll just take out one of those amino acids and have it line up nice and flat, and we'll see what happens. And by the way, I'm not going to go too much into what they found and all that. It's interesting, but it can be saved for another day when people are more technically minded and want to hear about it. But you will notice that here's the magenta is the barnase, and the other one is the streptokinase thing. And if you will look at it carefully, there is, in fact, a nice little loop-de-loop in both of them. The structure, both of them doing the same job, but with completely different amino acids. Well, not completely, but mostly different amino acids. Although they're different amino acid residues, they line up in almost exactly the same way. And that would be more obvious, actually, if you were to take this and look at it with something to shield your eyes so that you could only see one, and you made it small enough to where you could do this. This is not a good place to do it, but on paper, if you look past it, you can actually get a stereoscopic view, and you can see not only how it looks in the plane, but also it'll look like it comes up and down out of the paper. And those match as well. And now we come to the 2001, and this is the one that Axe did all by himself. Actually, he was still in Furst's lab, but Furst didn't want to put his name on this paper. Mutagenesis studies and alignments of homologous sequences have demonstrated that protein function typically is compatible with a variety of amino acid residues at most exterior non-active site positions. Remember this 1998 paper that he did. These observations have led to the current view that functional constraints on the sequence are minimal at these positions. Doesn't matter what you put in. Here it is shown that this inference assumes that the set of acceptable residues at each position is independent of the overall sequence context. 2 approaches are used to test this assumption. First, highly conservative replacements of exterior residues, none of which would cause significant functional disruption alone, are combined until roughly 1 in 5 have been changed. 1 in 5 of the whole molecule. This is found to cause complete loss of function in vivo for 2 unrelated monomeric enzymes: barnase, which is the one we've been doing, and a beta-lactamase produced by labeled TEM1 beta-lactamase. There's a bunch of other ones too. Second, a set of hybrid sequences is constructed from the 50% identical TIM1 and Proteus mirabilis beta-lactamases. So they just mixed amino acids from each one. Well, that should work, right? These hybrids match the TIM1 sequence except for a region at the C-terminal end where there are random composites of the two parents. All of these hybrids are biologically inactive. All of them. In both experiments, complete loss of activity demonstrates the importance of sequence context in determining whether substitutions are functionally acceptable. That is to say, you can get away with 1, you can get away with 2. If you do 20, you're in deep trouble. Contrary to the prevalent view, then, enzyme function places severe constraints on residue identities at positions showing evolutionary variability and at exterior non-active site positions in particular. Homologs sharing less than about 2/3 of the sequence identities should probably be viewed as distinct designs with their own sets of optimizing features. And it's not enough to put it in a protein family. You actually, if you're 2/3 identical, you're probably dealing with completely different proteins with their own sets of optimization. You can't go from protein 1 to protein 2. There is a barrier between the two different kinds of beta-lactamases that are virtually identical otherwise. Introduction. How amino acid sequences encode functional proteins has been a matter of intense interest for decades. Although many aspects of this problem remain unsolved, progress has been made in addressing the basic question of how tightly biological function constrains protein sequences. The currently accepted answer is that the constraints are quite loose, and Axe and company contributed to that. Any old, well, not any old, but 1/5 of them will work inside the core. Doesn't matter. Particularly positions on the exterior of a protein. So everybody thought, well, exterior, you got lots of room, you put whatever you like there. Particular positions on the exterior of a protein that are not directly involved in binding or catalysis. The origins of this low constraint view can be traced to 3 key developments in the latter half of the 1960s. During this period, the first systematic examination of a family of proteins, the globins, revealed both a high degree of sequence variation overall and a particularly high degree of variation at surface positions. Perutz et al., who I think got the Nobel Prize for some of this stuff. A subsequent study of natural hemoglobin variance in humans, Perutz and Lehman, accounted for the earlier finding by showing that exterior positions readily tolerate substitution while interior ones do not. At about the same time, a hypothesis which became known as the neutral theory of molecular evolution emerged, and notice Kimura. This theory proposes that the great majority of amino acid substitutions that become established over the course of evolution do so not as a result of selective benefit, but rather as a result of random drift. In order for this to be true, it must be the case that a sizable proportion of new substitutions have no significant functional effect. Mutagenesis studies where multiple substitutions are tested individually at each position along a protein typically show that most positions tolerate a variety of substitutions. Exterior positions, apart from those directly involved in function, usually exhibiting the most tolerance. You can put whatever you want, glutamic acid, arginine, tryptophan, well, tryptophan's one or two, but serine, whatever. And there's a whole bunch of references there. Result, we're skipping through. Testing for loose exterior constraints. Although this view seems to follow naturally from the data, it clearly involves an element of extrapolation. It implies that large-scale alterations of protein exterior should be functionally tolerated if active site residues and overall hydropathy are preserved, that is, attraction or resistance to water. Whereas the two principal lines of evidence upon which it based offer no direct support for this. A direct test of the corresponding extrapolation for protein interiors, where an entire hydrophobic core was randomly replaced by alternate hydrophobic residues, done by Axe et al., demonstrated that hydrophobicity is indeed the main criterion for a core to support basic enzymatic function in vivo. Okay. The original objective in the present study was to perform a similar direct test on exterior residues. The expectation being that extensive random substitutions should be even more readily tolerated, when initial results indicated that this is not the case. A more modest level of tolerance was considered. The revised objective was to determine whether exterior positions known to tolerate substitutions individually could be subjected to highly conservative replacements in mass without dramatically impairing function. How highly conservative? In order to avoid drawing conclusions that may be peculiar to one protein, two unrelated proteins were examined. Barnase, the single-domain monomeric ribonuclease used in the hydrophobic core studies, Axe's favorite, and TEM1 beta-lactamase, a two-domain monomeric enzyme that confers bacterial resistance to penicillin-like antibiotics. And of course, you can assess that by putting penicillin in various gradations into the medium and see how well this stuff grows. There are 84 exterior non-active site positions in barnase and 171 in TEM1 beta-lactamase. Obviously, TEM1 is a bigger molecule. Positions where change are tolerated have been identified in both proteins. In Huang et al. For the TEM1 and Axe et al. For the barnase. By applying biological selection to large populations of mutants carrying one or a few substitutions. Using this information, 5 groups of 5 replaceable exterior residues in barnase and 4 groups of 10 replaceable exterior residues in beta-lactamase were chosen for simultaneous substitution in the various places in the molecule. Residues in each group were replaced using the following conservative substitution set. Aspartic acid and glutamic acid could be interchanged. Nothing else. That is, they wouldn't put asparagine, they wouldn't put lysine, they wouldn't put just aspartic and glutamic acid. They only differ by one methylene group. Lysine versus arginine, which are both highly charged positive amino acids. Asparagine and glutamine, which are the amides of aspartic acid and glutamic acid. So those make a natural pair. Serine and threonine. Threonine has a methyl group on it. That's the only difference. Isoleucine and valine. Isoleucine has a methyl group on it. That's the only difference. Alanine and serine. Alanine goes to serine. They didn't put serine to alanine, which has a hydroxyl group on it. Leucine going to isoleucine, which is about the same size, and methionine going to leucine. So they're being really, really careful. Now, although these substitutions were chosen for chemical similarity, all except for asparagine and glutamine, are also the most common in nature for the same reason. The reduced natural frequency of asparagine and glutamine substitutions is explained by the fact that 2 DNA-based changes are required. All the other ones can be done with 1. After the mutations were induced into plasmids carrying the barnase genes, I'm continuing the paragraph, or beta-lactamase genes, Escherichia coli clones carrying the mutant plasmids were tested for the relevant activity by applying biological screens. Barnase, the one we've talked about, beta-lactamase to see how well it resists penicillin. The barnase screen simply indicates whether the cytoplasmic activity is above or below a toxic threshold of about 0.1% wild-type activity. All, that should be 5, I'm sorry. The computer does very interesting things, and I corrected most of them but missed one. All 5 single-group mutants of barnase have activities above this threshold. However, when double-group mutants representing all group pairs are constructed, most are found to be inactive. Since the double-group mutants bar MG, bar MB, and bar GB all retain function, the triple-group mutant bar MGB was constructed, but it was found to be inactive. So You know, you change some, you're okay. Change a little bit more, you're okay. You get past a certain point. This is kind of like, you see this 5. Well, I know what it is because I've had to change a bunch of them. But, you know, that's a mutant that if you needed that number 5, you'd have a hard time with. That's the one mutation there. However, some of them, if you put represent ong, you'd probably understand it, or UNG, or even PNG, you'd probably understand it. But you know, if you change the R to S and change the P to U and change, you know, if you after a certain point, you can't read it anymore. Mutant beta-lactamase activities can be quantified by determining maximum tolerable antibiotic concentrations. Again, single group mutants retain significant levels of function. Since the TIM1 enzyme has 2 domains, groups were combined so that all substitutions lie in one domain or the other. As with barnase, these double group mutants are more severely affected. The more you do, the worse it gets. The fact that a conservative replacement of only about 1/5 of the exterior residues causes near total loss of activity in both enzymes shows that function actually places very tight constraints on amino acid identities at the exterior positions. A quantitative estimate follows. And, a class B, A beta-lactamase from Proteus mirabilis has high degree of sequence similarity to the TIM1 enzyme, 50% amino acid identity, and 71% if you count similar ones. Amino acids. Both enzymes are highly active against penicillin G and ampicillin. To determine whether differing residues at aligned positions in these enzymes are comparably suitable, irrespective of context, a set of plasmids encoding 20 hybrid proteins was produced. The hybrids are identical to TIM1 enzymes except for a stretch of 78 residues at the C-terminus, where the sequences are random compositions of the TIM1 and Proteus mirabilis sequences. Because of the similarity of these parent sequences, the hybrids are about 90% identical to the TIM1 sequence. 3/4 of the difference from 3/4 of the differences from TIM1 occurring at exterior positions. Screening for in vivo activity as before revealed no detectable plasmid-mediated antibiotic resistance, meaning the activities are less than a very small percentage of the wild-type activities towards ampicillin and penicillin G, respectively. There's a whole bunch of other stuff they did. And the point that they're making is that, you know, you change enough and you get in severe trouble. Now, note that Douglas Axe was the sole author of the 2000 article. Nobody else wanted to be a critic of the feasibility of Darwinian evolution. This is saying you can't just pick any old sequence. For a protein with 90 external amino acids, conservative replacement may not be possible for more than 30 of them at the same time. I'm using the case of barnase, and probably less than 30. That means that 60 of them have to be left alone. Now, supposing one can randomly pick those 60, we don't know that yet, the rate of functional to total sequences would be roughly 20 to the 30 times 90 factorial divided by 30 factorial times 60 factorial divided by 20 to the 90th power, or 1 in 7 times 10 to the 54th. Now, that's probably not fair, because these were conservative substitutions. If one assumes that only 5 substitutions will work instead of the 1 or 2 that were tried most of the time, the result would be 1 in 2 times 10 to the 72. That's a lot. And we haven't factored in the core, which adds another 10 to the 4th or 5th or something like that. This is a much smaller target than anyone had anticipated. Axe's persistence in chasing this thing down paid off. Remember, he started with, well, does the core matter? No. Well, does individual amino acids matter? No, but he didn't quit there. He kept going. This, and his 2004 paper, which we'll discuss next week, is why Axe, hopefully not at quite this length, is why Axe is so confident that the design inference is valid. I think that the design intuition is in fact valid. But to be sure, I think it took someone, in this case Douglas Axe, to be willing to test it. And I've got to hand it to him. But that's my opinion. Now it's your turn. For those of you who can stay, that is. Comment down here. Go ahead. So, we've got a...
Oh, it's just a funny comment. Trouble in substitution, trouble with evolution.
Well, that's right. What it means is that in order to find an enzyme that actually works, and if you had all of the organisms known to have existed on Earth and you give them 3.8 billion years or whatever to work, that your chances of finding it are something like 1 in 10 to the 37th power, something like that. In other words, it ain't going to happen.
I'd like to make a theological comment, since I cannot make a scientific comment on this esoteric subject, but very, very good. It sounds convincing for a layperson when it comes to that subject. In the beginning, when God ended his work of 7 literal days, at the end of day 6, the scripture says, and God saw everything that he had made, and behold, it was very good. Everything would include the world of microbiology and enzymes and processes and substitutions. And when I compare that statement with this, I go in the direction of how I interpret very good, and you can take it for what it's worth. We often say the world was perfect.
But it doesn't actually say that. It's no,
I know in the Hebrew. And when you go to chapter 2, you can find an expression, not good. It's not good that man dwell alone.
Yeah, but God fixed that one.
So when something's not good, it's missing something. Very good means it has all the components necessary for continued existence and life. Now, I think you would agree with me that all of genetics and all of DNA is made so that there will be changes with time. There will be what we call mutations and many other changes, not just mutations. But at the same time, what I'm seeing is there's a whole system of protections, guards, to keep the changes from getting out of kilter. Now, I'm using lay language, and that's my take on it.
Yeah, no, I think you're right about that. And the other thing is that because you can put whatever core you want to into E. coli's barnase, and it seems to work fine, and if it's been millions of years, you really can't explain why it hasn't, why it's gotten to one wild type. If you start out with one man and one woman and God created it as the real thing, and he put the wild type at the pinnacle of perfection, then it is perhaps understandable why we would still have mostly wild type left, with a few mutations here and there, but 6,000 years is a whole lot less, or even 7.5 thousand years is a whole lot less than 6 million years, which is the conventional dating. And that's especially true if you had to get there from random mutations. There isn't enough time for the purifying effect. I mean, the only way you can do that is a founder effect. And according to standard evolutionary theory, there was never a founder effect because there were always thousands, maybe 10,000s of humans in existence. Whereas by the biblical record, there were only 2, and they were made that way. They were designed. So it makes a lot more sense that you have wild type now.
One more follow-up comment, and also it's based on Genesis 1, when it says that all of these things, whether plants or animals, whether vertebrates, invertebrates, they were all created after their kind. But one translation is that God just made lots of varieties rather than fixity of species. I think what you're showing here, all the work of Douglas Axe shows that we don't have fixity of species.
Well,
if you can extrapolate
I think
that.
That's unquestioned that we don't have fixity.
And
we certainly don't have fixity of subspecies. And,
yeah,
you know, if you want to go with Linnaeus, he said, well, maybe the species was a little higher up the chain than what he thought at the beginning.
could be.
And if you do that, you know, if you put the species at close to the family level most of the time, maybe at the genus level for humans, maybe that was a prejudicial assignment.
Mm-hmm.
But that then you can you can still say the fixity of species, and nobody's really going to argue with you that much in terms of having definitive data to disprove it, because the gaps between species kind of fade into each other sometimes. The gaps between genera can kind of fade into each other. But with a few exceptions, like songbirds, where it's probably the superfamily, the gaps between families is pretty much inviolate.
So what I'm seeing now, again, as a layperson, we have a tremendous resistance to change built into the genetics of every organism. But we also have a potential of change. And so there seems to be a balance there. We don't go the route with fixity of species, but we don't say there's wide open change where you can go from
Amoeba to man.
an amoeba to man. Yeah.
Yeah.
So that's my simple take on it.
I just might comment on this picture of change, since Warren raised it. If evolution had taken place, and I think you should see this probably in the microorganisms as well, you should have a tremendous number of fossils, of organisms trying to change who didn't make it.
Mm-hmm. You'd see the little limb buds, and then the longer limb buds, and then the longer
our
limb buds,
fossils-
and pretty soon you got wings.
Now, there's been changes, no question about it. But they fall into categories that we call phyla, or divisions, depending on what system you want to use. And the major changes between the phyla, we don't find those organisms out there.
But you don't find mouse bats.
Exactly. I mean, it's
The bats come in and they are ready to fly.
And turtles. Same thing, you know. Yeah, they're ready there with their pectoral girdle inside the rib cage, which
makes no sense.
Which is totally different for all other vertebrates. And so.
How do you migrate it to where it's right inside in the same line as the rib cage?
Yeah, and you come back to this work here. You raise the same question.
Well, here are these two penicillinases. They do the same job, but you can't get from A to B. There's this big wall there that just
you have to have certain specific changes to get the thing to work, and just at certain localities. You can substitute all these others here. You're still going to have that specific, which, I think you can't just say, well, sure, a lot of substitutions are fine, No, you have to have certain specific substitutions for an enzyme to work.
Yeah.
And all these other changes, very interesting that you can substitute all these. Not surprising, but you can substitute all these. But still, for evolution to take place, you're going to have specific changes at a specific locality in terms of the possible mutations and all this. This does not change the probability, I don't think.
And, I think that there's an important point here. Axe in his book says, trust your instincts. They're reliable. Well, how do you know they're reliable? Because he did the research. That's how. And what's really important on this is that you can't undo that research. I mean, I suppose you could accuse Axe of fraud, but nobody's done that in spite of the fact that he would be a nice, inviting target if he really hadn't done his homework. Just can you imagine somebody being able to show that Douglas Axe had it all wrong when it came to the structure of barnase, and how much variation it would tolerate. There would be people who would just love to do that, but nobody's done it because you can't do it. And this is important. You see, authorities are not authorities in science unless one of two things happens. They either have experimental data that they're passing on to you without coloring it, or they have, or perhaps both, or they have logic that appeals to you that you don't have to be the originator of it to be able to follow it. In which case, again, they're not the authority. It's the argument that's authoritative, and that scientific authority is most authoritative precisely where it's most transparent. Now, that's in some ways different from how some people perceive political authority. That is to say that in political authority, if you say, I want it this way, and everybody else wants it that way, and you get your way, then you have authority. But you can't do that in science. I would maintain also that you can't do that in theology. That, if you're the people who have the most authority in Christian theology are the people who most reflect the actual facts of Christ as they happened. And who most reflect, since Christ respected the Old Testament, the actual way that the Old Testament is read and should be read. That the less you get yourself involved in that, the better, if you want to be authoritative. Because what's really authoritative is the Word, not what you have to say. And I think that this is a really important thing. That's why Douglas Axe has more authority than 90% or 98% or whatever the percentage of it is of evolutionary biologists. Is not because I like him or that he has his heart in the right place or any of those kinds of things. It's because he is quoting stuff that if it's right, is right forever, and you can't change it. And the authority actually comes from the experiments themselves, not from Axe. And when you have that, then you can say, showing the logical progression, that you know what? You can trust your design instinct because it actually works.
It's the data that counts.
It's the data that counts. Anyway, yes, another comment.
I'm not as familiar with biology as I am with computers. I'm a computer programmer, and there's one thing that I keep thinking about, and that is that there's been a lot of research into the human brain, how it operates, and they say now that in a matter of minutes, there are new dendrites formed in the brain, new connections. So here we have a computer designed by God to rewire itself. If you want to represent that in terms of actual computer hardware, you're talking about a motherboard whose traces can move.
Yeah.
And create new chips on the motherboard.
Yes. Yes. And we can watch this happening, by the way. A person who has had a stroke and wiped out the part of the hemisphere that codes for speech, 5-10 years later, will sometimes reassemble on the other side the hardware that's needed to work for speech.
Right.
It had been referring everything all to the left side, which is typical for right-handers, and most of us are. And sometimes even left-handers have it on that side. But then you have a stroke, it wipes it out, and the brain says, yeah, but this is important stuff and we need to correlate it, and recreates on the other side a speech hemisphere.
Yeah, there was a child, I forget how exactly it happened, but there was a birth defect and most of the brain was missing. And eventually, as the child grew older, he got to where he could talk and he could function as a fairly normal child. And yet most of the brain mass was still missing. And eventually they discovered that the necessary functions for that to happen were in a very thin layer of brain mass that had formed on the inside of the skull, but most of the skull was still empty.
It's basically a hydrocephalus kid.
Yeah.
Yeah, no, that's happened. In fact, that has happened to a young lady I wrote about once who was a valedictorian of her class, and she had some kind of head injury, and so they did a CAT scan of the brain. And there's nothing there. But, I mean, she was obviously functional.
Yeah.
Well, it wasn't quite nothing there, but there was very, very thin layer, and somehow she'd been able to make that work.
Yeah, and my point is that
It'd be interesting to ask whether she was blonde or not. But
in the computer field, we have basically two classes of operating systems. One is based on Unix, the other is based on Windows. And in either system, if you change one bit, just a 0 or a 1, anywhere in a particular function, the whole thing breaks down. You might as well turn the switch off.
Yeah. Yeah. Well, there's some places where it doesn't matter, like a picture. You change one bit and you barely even see it.
Yeah,
but
I'm
there are
talking
other
about
places.
OS functions.
Yeah, but there are other places where you change a colon to a semicolon, and all of a sudden, depending on which language you're in, it no longer reads it, and you've been had.
Yeah. So not only can we not tolerate changes in the motherboard wiring, we can't tolerate even a tiny change in the code either.
Yeah. There are crucial areas where that's true. And the brain does better than that. It's amazing.
These authors should be sent to the Nobel Prize Commission. We should send them the book so they read it.
It would be an interesting gesture. I have a feeling that the opposition that Douglas Axe has little to do with the data and a lot more to where people wish the data were.
Of course. But not much opposition because he's going to become famous. Better.
He already is famous.